Hello and welcome back to another video. Today we will be solving Biology A-Levels 9700 Paper 42 February March 2016. So let's start with the first question. The rate of photosynthesis is affected by a number of environmental factors. Figure 1.1 shows the effect of light intensity on the rate of photosynthesis. Okay, so this is the graph of rate of photosynthesis on the y-axis and light intensity on the x-axis. State the limiting factor in the region A of the graph. So always remember whenever you asked about the slope region of a graph where the limiting factor is being asked, so the answer should always be that one factor which is on the x-axis. So here obviously it is the light intensity which is acting as the limiting factor. As you can see that light intensity is being increased, so the rate of photosynthesis is increasing. So the answer should be light intensity. Moving on, explain what is meant by the term limiting factor. So questions like these where you are supposed to define a term, it's better that you memorize the answer beforehand so that you are not losing marks in these type of questions. So you can begin the answer by writing that when a process is affected by more than one factor, the limiting factor is the one that prevents any further increase in the rate of the process. So remember, a process that has more than one factor, right? And the limiting factor would be the factor that is mainly inhibiting the process, just like light intensity here. Part 3. Explain why there is no further increase in the rate of photosynthesis beyond point C. Now, as you can see, beyond point C, the rate of photosynthesis has become constant, right? It has become constant, meaning, always remember when this happens, you have to say that the limiting factor which is on the x-axis is no longer acting as the factor which is on the x-axis is no longer acting as a limiting factor. There must be another factor which has become a limiting factor. And also give an example of what factor it could be, since you have studied about different factors that affect the rate of photosynthesis. So you can write light intensity is no longer acting as the limiting factor It could be the carbon dioxide concentration or temperature. Okay, moving on. For many plants living in temperate regions, the optimum temperature for photosynthesis is approximately 25 degrees Celsius. Suggest reasons why the rate of photosynthesis decreases at temperatures above 25 degrees Celsius. So we know that in light independent stage, in the Calvin cycle, there is an enzyme which is used named rubisco that fixes the carbon dioxide. So at high temperatures, we know that enzymes denature. So that rubisco enzyme would denature. So let's write down this point. At high temperatures, due to which less Calvin cycle and less carbon dioxide is fixed. Hence, this shows less rate of photosynthesis. Or you can say decreased rate of photosynthesis. Other than this, we also know that when temperatures increase, more transpiration happens. And when more transpiration happens, there is a danger of wilting. So a plant releases the stress hormone and it makes the stomata close, right? So if the stomata are closed, there would be reduction in carbon dioxide uptake, due to which the rate of photosynthesis would also decrease. So let's write this down. Means increased transpiration, leading to uh, resulting in reduction in carbon dioxide uptake. And if there is not in much carbon dioxide present, the light independent reaction would be lesser, so rate of photosynthesis would be decreased. Moving on to question number 2. The IUCN Red List provides information about the conservation status of species throughout the world, including the American badger, Tuxedia taxus, and the black-footed ferret, Mustela nigrives. Figure 2.1 shows an American badger, and figure 2.2 shows a black-footed ferret. Okay. Figure 2.3 shows the IUCN conservation status of the American badger and the black-footed ferret in 1987 and in 2013. Okay, let's move on to the question. American badgers and black-footed ferrets are both predators. American badgers feed on prairie dogs and a range of other animals. Okay, so they have quite a few animals that they feed on. Black-footed ferrets feed almost entirely on prairie dogs. Almost entirely. Uh, American badgers do not have any animal predators. Black-footed ferrets are preyed upon by American badgers and several other predators. Suggest reasons why black-footed ferrets are an endangered species, but American badgers are not. Well, this is pretty simple. You can go according to these bullet points and proving why that this is the case. You can firstly say that ferrets have only one kind of prey, but American badgers have a range of other animals that they feed upon. So you can say that first, according to the first bullet point. Ferrets And in the reverse argument, you can say that American badgers feed on prairie dogs and other different animals. Second bullet point is black-footed ferrets feed almost entirely on prairie dogs. That we can say that if the number of prairie dogs decreases, that means the number of ferrets will also decrease. But we cannot say that if the number of prairie dogs decreases, the number of American badgers would also decrease. So that is another factor which is contributing to the species of black-footed ferrets being endangered. So we can say that reduction in dogs population will lead to decrease in number of ferrets. Other than that, we also read that American badgers do not have any animal predators, while black-footed ferrets have many predators. And in the reverse argument, you could say badgers do not have predators. Part B. In 1978, the world population of black-foot ferrets consisted of only 18 animals living in captivity. A number of different agencies worked together to prevent the extinction of this species. Their goal was to produce young black-footed ferrets to be released into the wild. The survival and the breeding of the animals in the wild would then be monitored and supported. The collaborating agencies included local government, universities, zoos, native tribes that owned undeveloped reservation land. Outline how these different agencies could contribute to successful conservation of the black-footed ferrets. Okay, so this is a 3 marks. So let's consider first the local government. So what does a government do? We know that one of the things they do is passing laws, right? So you can say that local government authorities should pass protection laws for the conservation of the black-footed parrots. So that would contribute to the conservation. So local government authorities So universities can carry out research in this department because commonly there are a lot of equipments found in universities, so that would help for the research, and it would be good that you give examples for what researches could be done. So universities carry out researches, and those researches could be maybe regarding how to improve the success of breeding program, or it could be that they perform IVF, which is the procedure where you remove an egg from the ovaries from a female and then they are fertilized with the sperm in a laboratory. So perform. This could be done because there are several research labs in universities. And like this, you can even write about they could monitor genetic variability, coordinate with student records, and they can also, determine suitable habitat requirements for release sites and monitor wild population. So these are all stuff that could be done within a research by maybe a group of students. The third agency is zoo. Now, the best place where you can keep an animal in captivity would be a zoo, right? So, zoo is the main area where the captivity program could be held. So, zoos. Now, since the native tribes are the ones that own the undeveloped reservation land, they could be maybe contacted to provide that suitable land for the successful conservation of the ferrets. Part C. Black-footed ferrets were released at three different locations in the wild at different times. Each population was established from captive bred animals. Figure 2.4 shows the population size of black-footed ferrets at three release locations. Population sizes. Okay, so we have Arizona, Wyoming, and South Dakota. Describe the patterns of population growth at three locations where black-footed ferrets were released. 3 marks. So the main thing that we see about South Dakota is that there's a very, very steep increase in the population size. So that's right down. Um, South Dakota showed a steep increase other than that. Wyoming, here, see this? Wyoming has a constant population growth and then it is decreasing and then increasing. So that's definitely something that we should write about. Is constant in Wyoming initially, then it decreased. Here you can read the reading. It decreased to approximately, wait, approximately 10. Actually, yes, 10 number of individuals. So it decreased to 10 number of. And then from the graph, you can see that for Wyoming, after decreasing, the population size has increased again here. So let's write that down. For Arizona, you can see that the population size has been really low and then it has increased a little bit. So you can say that Arizona population was very low initially and then it recovered or it increased. You can even give the figures. You can even give the span of the years for which it was less and then it increased. So essentially, whatever point you give, if as long as it is valid, you'll get a mark. Table 2.1 shows information about the gene pool of the populations of black-footed ferrets and the leg sizes of the black-footed ferrets at each release location in 2004. So we have the leg sizes and we have the gene pool. All three populations were started by animals from the same captive population. In this original captive population, 100% of the genes surveyed showed a polymorphism, that is, that they had more than one allele. The mean number of alleles per gene locus was 2. The population at the South Dakota location in 2004 maintained the same level of genetic variation and leg size data as the original captive population. But the populations in Wyoming and Arizona showed changes. So Wyoming and Arizona, okay. Use table 2.1 to describe how the gene pools and leg sizes of Wyoming and Arizona black-footed ferret populations have changed compared to the original captive population. Since we know that the original captive population is similar to South Dakota, right? So we can use South Dakota as a standard, as a reference to which we will be comparing the Wyoming's and Arizona's readings or percentages, right? So let's first see for Wyoming. You can see that the percentage of genes that are polymorphic has decreased, right? It has decreased, and here you can also see the mean number of alleles per gene locus for Wyoming. Wyoming has decreased. So one thing we can write about this is that has lost polymorphism, or you can even say that Wyoming has lost alleles or genetic variability, because polymorphism gives genetic variability, right? So you can even say that the gene pool has decreased for Wyoming because of the lost alleles. So as long as you're writing any of those, you'll get a mark. Now, comparing the Arizona's percentage of genes that are polymorphic, you can see that it is constant, and here the main number of alleles per gene locus has increased. So just like what we wrote for Wyoming, we should be writing in opposite terms that Arizona has gained maybe genetic variability, alleles, or polymorphism, or the gene pool has increased, whatever you want to write. Now let's see the mean length of lower back leg. For South Dakota to Wyoming, it has decreased, while mean length of lower front leg has also decreased. So we can say that uh, and if you compare with Arizona, they are constant. So there's no change in the bone length. Moving on. With reference to figure 2.4, suggest reasons for the changes you have described in part 2. So first of all, let's consider this point here, that Wyoming has lost polymorphism, or we said that Wyoming has a smaller gene pool. Now, the reason for this would be if you go back to the graph which is given in figure 2.4, here you can see that the population size of Wyoming versus South Dakota, there is a huge difference between them. So, if the population size is small, then obviously the gene pool would also be really small, right? So, you can write down. Now, let's see. We also then gave this point right here that Arizona has gained polymorphism. What's the reason for this? So you can see that Arizona's population size is even smaller than Wyoming's in comparison to South Dakota. So obviously the reason of its increased polymorphism cannot be that its population size is higher. So it must be due to some mutation, right? Some random mutation. So and the third point that we wrote was that Wyoming's ferret bone lengths were smaller. Now for this, whenever you think about bone length, you think about that it is being affected by the environment, right? So a reason for it to be smaller could be that there is less food available, or maybe there is inbreeding between the ferrets, due to which that also inbreeding could be said because the ferrets population in Wyoming is very less, so there must have been some inbreeding, due to which those alleles which code for a lesser, which actually results in a lesser length of the bone, actually they should have been expressed. So inbreeding could be one cause, or maybe less food available, due to which the ferrets have decreased bone length. So Wyoming's, sorry. Part D. In 2008, some black-footed ferrets were born in captivity as a result of IVF using frozen sperm that had been stored for several years. Explain the benefits of using frozen sperm in captive breeding programs. Now, if you have is frozen sperm, there is an increased number of potential mates for it, right? If you have the sperm with you, you can mate it with any of the female's eggs. So the first point could be that it increases. And if the number of potential mates is higher, then that means the gene pool would also increase. So it is also possible to transport the sperm to different facilities, maybe to zoos and breeding facilities. So now frozen sperm is acting as a gene bank. What is a gene bank? It may be a collection of seeds or plants or animals. Their genetic material is preserved, typically to maintain genetic diversity. Frozen. sperm acts as a gene bank. We wrote here that it will allow the gene pool to increase, so we do not need to write again that it is to preserve genetic diversity. One more benefit would be that if there are animals that have died, we can store their alleles. So maybe there are some desirable characteristics, so their alleles can be stored. So that's it. Question number 3. Myofibrils in striated muscle consist of contractile units called sarcomeres. When an impulse stimulates striated muscles to contract, calcium ions are released from the sarcoplasmic reticulum. Describe how the release of calcium ions leads to contraction of a sarcomere. Well, look, this question is very simple, and it's really, really important that you remember these steps. So, firstly, the calcium ions are released from sarco. Now, these calcium ions bind to troponin. Now, this troponin is going to change shape. Troponin will make the tropomyosin to displace from its position because tropomyosin was initially covering the myosin binding site. So when troponin changes shape, it causes the tropomyosin to expose that myosin binding site. So the myosin head, which has the ATP, will now bind to actin, pulling the actin with it, right? And that will cause the sarcomere to contract. So part B, the many-branded crate is a venomous snake. Figure 3.1 shows a many-branded crate. The venom from the many-branded crate contains bungarotoxin. In mammals that are bitten by this snake, the venom acts at the neuromuscular junction. So what happens at the neuromuscular junction? First of all, there are two types of neuron, and the presynaptic neuron and the postsynaptic neuron. And we know that the acetylcholine is released from the presynaptic neuron towards the postsynaptic neuron. The acetylcholine is basically responsible for attaching to the receptors at the postsynaptic membrane of the neuron, postsynaptic membrane, and that causes the influx of sodium ions. Sodium ions are diffused into the cytoplasm of the postsynaptic neuron. Now, the sodium ions are obviously going to cause depolarization of the postsynaptic membrane, and then the electrical impulse can continue down the axon of the postsynaptic neuron. So, if this snake has bitten a mammal, then how could this muscle paralysis could be caused? First of all, since the first point we said about the acetylcholine was that since acetylcholine is a neurotransmitter and it is released from the presynaptic neuron, we can say that this venomous snake could cause the reduction in the release of acetylcholine by the presynaptic neuron. So first point, I'm writing acetylcholine as this, its short form, or you can even say neurotransmitter. Then normally acetylcholine binds to the receptor on postsynaptic membrane, so the venom may cause inhibition of the binding of acetylcholine on postsynaptic membrane, so prevents. Okay, and if the binding is not occurring, then there would be no depolarization of the postsynaptic membrane. And hence electric impulse is not transmitted. Other than this, we also know that it is important that acetylcholine is broken down to avoid permanent contraction of a muscle or permanent depolarization of the postsynaptic membrane. So the venom of the snake can also inhibit acetylcholinesterase, which is the enzyme that breaks down acetylcholine. So remember that this enzyme breaks down acetylcholine, and if it is not present, so acetylcholine would not be broken down. And if acetylcholine is not broken down, then the permanent depolarization of postsynaptic membrane will happen, and that will also cause the muscle paralysis. Okay, so this question is on gametogenesis, and gametogenesis in humans and plants has been removed from the syllabus for 2022 to 2024. So we are not going to do this question. Okay, so figure 4.1 shows a Komodo dragon. Okay, this species of lizard is only found on 5 Indonesian islands. In 2006, 2 captive female Komodo dragons A and B in British zoos each produced healthy offspring, 1 to 4, despite never having mated with a male. Okay. Female B was later mated with a male Komodo dragon C and produced another offspring 5. Okay. Genetic analysis was performed on the 2 females, their offsprings that had no father, the male C, and the offspring 5 produced by mating between B and C. The results of genetic analysis are shown in the table 4.1. Different alleles at 4 gene loci P, Q, or and S could be distinguished by their different lengths. The alleles for each gene are shaded differently. In table 4.1, the sex chromosomes in this species are called W and X. Okay. Using table 4.1, identify the gene locus that shows most genetic variability and name the process that gives rise to different lengths of the allele. Name the process that gives different lengths of the allele. So let's see. Most genetic variability. So as you can see that the most genetic variability is shown by at the gene locus R. You can see that here it is 188 base pairs and then 206 here. So there's quite a few, quite some difference. And the process that would give the genetic variability process would be mutation. Using table 4.1, state and explain which animals are heterozygous at one or more of the gene loci. So for this, what you need to do is that look at them turn by turn. So let's see for female A first. It is heterozygous at locus B, since these are different, the base pair number. So yes, heterozygous at locus B. Yes, heterozygous at locus Q as well, R as well, but not S. So female A is heterozygous at PQR. Let's look at the offspring from female A, offspring 1. So it's not heterozygous at any locus. What about offspring 2? It is also not, it is homozygous at all loci. Female B, heterozygous at P and at S. So female B offspring of female B, not heterozygous at any locus. Not heterozygous at any locus. Male C is heterozygous at locus P, Q, R, not S. Let's look at the offspring 5. It is heterozygous at P, R. Okay, so P and R. Okay. Clones are organisms or cells that are genetically identical. Students made suggestions about the Komodo dragon offspring 1 to 4 produced by a female that had never mated with a male. Offspring 1 and 2 are clones of each other, and offspring 3 and 4 are clones of each other. Okay. Offspring 1 to 4 were produced by asexual reproduction using mitosis only. Explain with reference to specific loci whether the data in 4.1 support or do not support these suggestions. Offspring 1 and 2 are clones of each other, and offspring 3 and 4 are clones of each other. So let's see. You can see that offspring 1 and 2 are not clones of each other, because here it is 216, while here offspring 2 is 211, right? Again, 151 and 154, different, 200, 188. And this is the only gene loci where the offspring 1 and 2 have same base pairs, right? So it means that they're not clones of each other. So they're not clones because offspring 1 and 2 differ at loci P, Q, and R. Let's look at offspring 3 and 4. So they differ at P, and they also differ at S. So moving on, offspring 1 and 4 were produced by asexual reproduction using mitosis only. So first of all, keep in mind the principle of asexual reproduction. So whatever the offspring is produced, it is identical to the parent, right? So let's see if the offsprings are identical to their parents. So offspring 1 and 2 should be, according to the statement given, they should be identical to female A. But let's see if they actually are or not. So you can see here that offspring 1 is different to the parent, which is female A at B, and here the offspring 2 is different from female A. So gene B, they are different. They are also different. So Q as well, and different, different, and these are the same. So let's see for offspring 3 and 4. This is different, different. These are the same. These are also the same. Different and different. So P and S. Reproduction without a male has evolved in a number of species that live on islands. Suggest advantages and disadvantages of this type of reproduction in an island habitat. So, island. So let's first look up the advantages. Since it is an island that is being discussed here, so the island must have a small population of any species, right? So the mates must be scarce. So if this ability is evolved where the reproduction is done without a male, it will be beneficial. So we can write that. Females that females can still reproduce without male continual population. Also, if you noticed here in this table that when you look at the offsprings produced by females that have reproduced alone, you can see that their genotype is for the sex chromosome is double Z, right? Which shows that all the uh, all the offspring that were produced by the female when in the absence of a male were males. You can see that. And how would you know that the genotype for male is double Z? You can see that this male right here, C, has a genotype of double Z. So you know that from there. So if we know that the offspring produced by a female reproducing alone, offsprings produced are males, right? So if offsprings are all male, so female could then mate with them to keep the population going. So offspring. Also, reproducing without a male also ensures that uh, the alleles that the females have, which are needed to survive in that habitat, they are passed on, right? So retains adaptations for that environment. Okay, so now the disadvantages. Disadvantages may be that um, maybe yes, that there would be reduction in genetic variation. And you can say that gene pool would be smaller because of the small population. One more very important disadvantage would be that heterozygosity would be decreased, and this could be seen from the table here, that in offspring 1 and 2, it's mostly homozygosity, like these two are the same here as well. And then you look at 3 and 4, they're also pretty much all the same, right? But if you look at offspring 5, which was a true female B and male C, you can see that it shows heterozygosity. So you can conclude that heterozygosity and what does happen when heterozygosity will be lesser? It means that harmful recessive alleles may come together. There's a high chance of them coming together. Another point here to be added is lack of hybrid vigor. Now it's important for you to understand what exactly is hybrid vigor. So let me write down the definition too. It's basically the tendency of a crossbred individual to show qualities superior to those of both parents, right? But here only one parent is involved, so obviously the qualities would be lesser. They would be lesser superior, right? So the tendency, and with the help of these qualities, what do they do? They help the individual survive climatic changes. But if these qualities are not present, obviously they are more likely to perish. And as I explained, that they would not be able to adapt to changing environments. And that is it for this question. Question 5, part A, figure 5.1 outlines how two hormones A and B are involved in the regulation of blood glucose concentration. Okay, with reference to 5.1, name the control mechanism that regulates blood glucose concentration, and that would be negative feedback. Hormone A. So this is the hormone A, and it is acting on the liver cells to release glucose. So it must be glucagon. The enzyme and glycogen phosphorylase are both involved in the formation and breakdown of glycogen in the liver. Figure 5.2 shows the activity of the two enzymes in the liver after consumption of a glucose meal. Okay, describe and suggest an explanation for the changes in the activity of enzymes, glycogen synthetase and glycogen phosphorylase. So it is safe to say that after intaking a glucose meal, the blood glucose it is safe to say that after intaking a glucose meal, the blood glucose concentration must be high. So and we know that when the blood glucose concentration increases, insulin is released and more glucose enters the liver cells. Right. Now, this leads to increase in the activity of glycogen synthase, because glycogen synthase actually catalyzes the T step of synthesizing glycogen, right? So, and you can see in this graph that it is increasing uh, after the glucose meal. Also, it is important that you know what the function of glycogen synthase is. So remember that it catalyzes the key step of synthesizing glycogen. Also, this process would be glycogenesis. Now you have to also point out that the activity of enzyme glycogen phosphorylase is decreasing as the time increases after the glucose meal consumption. So the and due to this glycogen, if there's a decrease in the activity of enzyme glycogen phosphorylase, that means there is decrease in glycogenolysis, which is actually the breakdown of uh, glycogen to produce glucose. So since the person has taken a glucose meal, so obviously he does not need extra glucose. So there is a decrease in the process of glycogenolysis, which is performed by the enzyme glycogen phosphorylase. So know the difference between glycogenolysis and glycogenesis.